In 1988, a selected committee of the US National Academy of Sciences defined the initial aim of the HGP, the Human Genome Project, which was to sequence the entire human genome as well as the genome of certain selected non-human model organisms like a mouse, rat, fruit fly, roundworm, baker's yeast, and the common intestinal bacterium E. coli. Think of it as deciphering a mysterious code that is the instruction manual to humans, or better yet, like figuring out the ingredients that make up a really good cake. Except this is like a million times better easily because you can't cure cancer with the ingredients of a Victoria sponge. Now, unfortunately, we haven't actually cured cancer yet, but the good news is that the widespread availability of affordable genome sequencing has fueled a surge in genomic research across various fields such as genetics, genomics, epidemiology, and drug discovery. For instance, the International Cancer Genome Consortium and the Cancer Genome Atlas are undertaking large-scale genomic data collection and analyses for numerous cancer types. Researchers then aim to identify disease-causing variants and establish well-founded correlations between sequence variation and specific phenotypes, improving our understanding of inherited diseases and helping us develop genetically targeted medicines. However, discoveries from the HGP have already paved the way for several successes like the cloning of genes responsible for certain diseases. Genomic maps also serve as tools for research. But what I really want to zero in on is something called comparative genomics. In this context, that would mean us studying the genome of a low organism, which in turn provides insight about our own. It's kind of like doing a practice paper before an exam. Are the same questions going to be asked on the exam? No, but does it help you perform better on the actual exam? Yes. Animal models are selected for research because of their similarity to humans in terms of genetics, anatomy, and physiology, in addition to their unlimited supply and ease of manipulation. It's easier for scientists to use large numbers of animals rather than people to attain conclusive results. Here's a fun fact for you. Over 95% of all existing transgenic animals are mice, specially made to serve as models for human diseases. In 1984, the very first oncomouse was produced by inserting the MYC oncogene into the mouse genome to predispose tumor growth in the animal. In retrospect, not such a great day for the mouse, but for us, that day marked a new chapter in the field of biomedical research and changed our scientific understanding of disease and disease treatment forever. Nonetheless, today, over 1,000 mutant strains exist as models for inherited genetic diseases. They're made by mutating the same gene in mice that is responsible for the human condition in approximately 100 genes. Diseases for which mouse models exist include several types of cancer, heart disease, hypertension, metabolic and hormonal disorders, diabetes, obesity, even psychiatric conditions like anxiety and depression. The list really does go on. Therefore, this allows researchers to study the effects of these mutations and test potential treatments in a controlled environment. By leveraging genetic manipulation techniques, researchers can accelerate the drug discovery process and bypass some of the trial and error steps associated with traditional research methods. This by far is the greatest consequence of the Human Genome Project. Its revolutionary effect on the course of biomedical research, as well as fundamentally altering our approach to understanding and treating diseases in ways never thought possible till now. And although there are certainly questions raised about the ethics of it all, many believe that as long as decided ethical limits are adhered to by all, the pursuit of answers to crucial scientific enigmas must be our first objective. Because maybe the cure to something as insurmountable as cancer lies in the genetic strands of a tiny white laboratory mouse. Thank you.